Hi, my name is Craig Smell, and this is my final project presentation on rituximab and gene MS4A1. Rituximab is a genetically engineered human murine monoclonal antibody. The drug targets a B-cell cell surface antigen known as CD20, a protein which is coded for by the gene MS4A1. Approved by the FDA in 1997, it was the first monoclonal antibody approved to treat malignancy. Rituximab is indicated for certain forms of non-Hodgkin's lymphoma, leukemia, rheumatoid arthritis, and dermatological conditions. Chemotherapy is usually co-administered with rituximab. Rituximab is delivered intravenously, has a low volume of distribution, a half-life of 76 hours, and is excreted via immune response. Its effect is to kill B cells by binding with CD20 to initiate an immune response, as well as changing the B cell intracellular environment. Several adverse effects have been observed in patients treated with rituximab, including response to cell breakdown products and loss of immunity to certain viruses. The incidence of these adverse effects vary considerably. Rituximab is thought to promote cell death in three ways, which can be categorized as A. Eliciting an immune response, and B. Changing the targeted B cell intracellular environment. The drug elicits an immune response comprised of anti-murine antibodies and direct cell death signaling. Within the cell, an increase in sensitivity to apoptosis means that cells become more sensitive to chemotherapeutic drugs. MS4A1 is a gene with 6 exons located on chromosome 11. CD20, the protein product of MS4A1, is involved in B cell activation and proliferation and regulates calmodulin-dependent protein kinase. However, the exact functions of this protein are yet to be fully elucidated. CD20 is expressed on normal mature B cells and around 80% of mature B cell neoplasms, but is not expressed on stem cells, pro-B cells, or terminally differentiated plasma cells. This allows plasma cell antibody production to be maintained, at the same time allowing early-stage B cells to develop into their normal form, thus mitigating immunosuppression caused by rituximab. Binding of rituximab with CD20 results in increased sensitivity to apoptosis. With CD20 blocked, phosphorylation of an SRC family tyrosine kinase is blocked, which initiates a cascade eventually leading to downexpression of BCL2, which results in increased sensitivity to apoptosis. Additionally, via a separate pathway, rituximab results in the upregulation of RAF1 kinase inhibitor protein, which blocks two signaling pathways both involved in expression of BCL-XL. The loss of expression of this gene results in a decrease of the apoptosis threshold and, as above, increases sensitivity to apoptosis. Variation in the MS4A1 gene observed to date includes single nucleotide polymorphisms and a frameshift mutation resulting from a one base pair deletion. MS4A1 variation in non-disease individuals is not well studied. 509 single nucleotide polymorphisms have been identified in this gene, 57 of which have been mapped to coding regions. There is allele frequency data on most SNPs available from NCBI, but almost no functional annotations exist. In a study of a patient who developed resistance to rituximab after first dosage, a frameshift mutation resulting from a one base pair deletion was found in the C-terminal region of CD20. The C-terminal region was later found to cause the peptide to adopt a cyclic conformation essential for rituximab binding. Also, the researchers demonstrated that the mutant allele was upregulated compared to the wild type at time of relapse. An association study of 164 Chinese CD20 positive non-Hodgkin lymphoma patients identified three SNPs of interest in response to rituximab therapy. Those homozygous for the ancestral allele C at one of these SNPs showed significantly greater complete response rates than those patients heterozygous or homozygous for the derived allele T. Conversely, the conclusions of a separate study suggested that no genetic variation is responsible for failure of rituximab therapy in some patients. However, the analysis was limited a priori to include sequencing of exon 5 only, as it was known to the researchers that this region codes for the rituximab binding region on CD20. However, as was explained above, the cyclic conformation caused by the C-terminal was shown to be essential to rituximab binding, and the C-terminal region is not coded for by exon 5. Additionally, the authors used the antibody L26, the most common antibody in use against CD20, which was later shown not to be capable of detecting all mutations. Rituximab is a drug that could benefit from pharmacogenomic insight, identifying patients who are likely to become rituximab resistant or may be resistant from first dose helps formulate treatment strategies. Given that the cost per infusion runs at between $5,000 and $10,000 in the US, it's important to allocate the resources to the treatment with the greatest chance of success in each patient. Immediate research efforts could focus on mapping the C-terminal frameshift deletion to the reference genome sequence in order to sequence this region in other patients to determine the prevalence of this variation in an effort to explain observed cases of rituximab resistance. This presentation is focused on pharmacodynamics of the drug, but there is also evidence that genes involved in the pharmacokinetics of the drug are important also. Finally, the implementation of pharmacogenomic warnings for rituximab should take into account already complex treatment guidelines. This concludes the presentation. Thanks very much for listening.